I think we are going to start, so I'm going to change to Spanish and then for the introduction. Okay. Okay. Hola, buenas tardes a todos. Bienvenidos al primer seminario de este semestre del programa de Frontiers in Genomics, que es parte de la licenciatura de Ciencias Genómicas de la UNAM y que está a cargo del Centro de Ciencias Genómicas y del Instituto de Biotecnología de la UNAM. Como saben, las preguntas del seminario son siempre al final, y ya sea que las puedan poner en el chat o levantar la mano en la pantalla y entonces preguntar a ustedes directamente. Bueno, como les decía, iniciamos esta serie de seminarios con algo un poquito diferente a la mayoría de los seminarios del programa, pero va a ser muy interesante, van a ver, y nos va a hacer pensar muchas cosas de qué vamos a comer después de la plática. Bueno, para mí es un gusto presentarles al doctor Ahmed El-Sohemy. El doctor Sohemy es profesor y chair de la Universidad de Toronto y tiene la cátedra de Canadá en Nutrigenómica. También es fundador de una compañía muy interesante que se llama Nutrigenomics, Inc., en donde es el jefe de la oficina científica y el chair del Advisory Board internacional de la misma compañía. El doctor Sohemy hizo su doctorado en Ciencias de la Nutrición en la Universidad de Toronto y después realizó una estancia postdoctoral en la Universidad de Harvard. Como ya los ven en el título, el objetivo de sus investigaciones es entender cómo diferencias en nuestros genes responden a la dieta y así obtener un mejor desempeño y una salud óptima también. Ahmed ha publicado más de 180 artículos científicos y dado más de 250 conferencias por invitación. Es miembro del comité editorial de 10 revistas científicas y es revisor de otras 30 y de agencias que dan donativos. Y es editor en jefe de la revista Genes and Nutrition. Es o ha sido asesor científico en diferentes paneles institucionales en Canadá y y en otros países. Y el doctor Sohemy tiene varios premios muy relevantes, entre los que voy a mencionar solamente algunos, como el Centrum Foundation New Scientist Award for Outstanding Research by the Canadian Nutrition Society, the Mark Beaver Professional Award by the American College of Nutrition, and the Stalin Watch Award by the American Nutrition Association. Well, with that, I welcome Dr. Sohemy. Thanks again to be here and participate in this program. And now we are ready to hear your lecture.
Great. Thank you very much, Dr. Zurita, for the kind introduction. Hello to everyone online. As I mentioned earlier in the talk yesterday, of course, it would have been wonderful to have the opportunity to be there in Mexico in person to present this work and to meet with all the faculty and students, but obviously given the circumstances, that was not possible this year. Hopefully, perhaps in a couple of years, there'll be an opportunity for another set of seminars with an in-person meeting. So today what I wanted to do is present to you some of the research that we've been doing at the University of Toronto in the field of Nutrigenomics or nutritional genomics, and looking to see how genetic testing could be used to provide personalized nutrition advice. And so first, by way of disclosure, I am the founder and I do hold shares in the company, Nutrigenomics with an X, which does provide genetic testing for personalized nutrition to healthcare practitioners. And I will touch a little bit on the company just at the end of my talk to describe how we've translated some of the discoveries from the laboratory into practice. So first, for those who might not be familiar with the term nutrigenomics, as you can gather just by the breakdown of the word, it really is the branch of nutritional sciences that deals with the interface between nutrition and our genetic makeup, with one of the ultimate goals being looking to see if and how we can use genetic testing to understand and determine why some individuals respond differently from others to the same foods, beverages, and supplements that they consume. Now, from a research perspective, we can think of two sides of a coin. On the one hand, we know diet can affect genes. Nutrients can upregulate or downregulate the expression of various genes. Nutrients can also cause epigenetic modifications that can ultimately be inherited by subsequent generations and have important impacts on various phenotypes. The flip side of this coin, illustrated by the bottom arrow, is really understanding how variations in genes affect the way we respond to diet. And so this is really the focus of our research program and the focus of the work that I'm going to be presenting today. So genes influence, they encode for proteins that affect the absorption, the metabolism, the distribution, the uptake, and breakdown of virtually everything we ingest. But another element of this bottom arrow is before a gene can even act on a nutrient, genes can also influence the foods that we select. So genes influence our likes and dislikes of various foods. And this is an area that we got interested in several years ago. And it was really a chance discovery that we made. So one of my former PhD students at the time, Karen Enney, she's currently the director of clinical services for the company Nutrigenomics. But during her doctoral research, she was looking to see whether or not we can identify genetic variants that can explain differences in an oral glucose tolerance test to see if people who consume the same amount of glucose, if those genetic variants can explain differences in the area under the blood glucose response curve. And we focused on the glucose transporter type 2, Glute2, because it seemed like it was a good candidate. It was expressed in the pancreas, and it does detect rises in blood glucose and causes insulin to be secreted and clear that. But we didn't see any differences really in the area under the blood glucose response curve. What we did find just by chance was those who had a particular version of the Glute2, it causes an amino acid substitution. Those who have the isoleucine allele consumed more sugar on a daily basis. Just habitual sugar consumption was greater in comparison to those who have the threonine version of it. So we demonstrated this in 2 different populations, and this was really kind of an exciting finding. We reported this in Physiological Genomics, and it was the first time a gene was identified that explained an eating habit. And in this particular case, it was sugar consumption. So part of Karen's doctoral thesis involved looking at other potential candidate genes. And then she identified a variation in the TAS1R2, which is a sweet taste receptor that's expressed on the surface of the tongue, and showed that individuals who have a particular version of this also consume more sugars on a daily basis. Now, you can imagine it's quite possible that some people have both of these polymorphisms, and that makes them consume even more sugars, the highest amount of sugar. So when we reported this, it was the kind of thing that got some media attention. This was the headline that was on the front page of the Toronto Star, the largest newspaper in Canada, and it said, "Craving something sweet, blame it on your DNA." Not quite the message that we had wanted to communicate, but this is what sells newspapers. And it got some interest from a number of groups. And one of them was a colleague of mine in the Faculty of Dentistry who saw this in the newspaper and contacted me and said, well, we know that sugar is a risk factor for dental caries. He was curious to know if individuals who have these polymorphisms are at a higher risk of tooth decay or having dental caries. So we collaborated on a project, and Tabitha Chang, who was a dental student, conducted a number of examinations on the patients, and then we genotyped them to look to see which version of the Glute2 and TAS1R2 they had. And we found that those who had the combination genotype that's illustrated by the purple bars, these are the ones that are associated with the highest level of sugar consumption. They had more than twice the number of decayed, missing, or filled teeth, DMFT. And this is a recognized scoring system. It was also confirmed by X-ray and also using an international scoring system. So these are subjects that are only in their 20s, and already, because they have these genetic variants that predispose them to consuming more sugar over the course of their lifetime, by the time they get into their 20s, they already have twice the number of dental caries. So clearly it has implications for oral health. Since we published this back in 2013, other researchers replicated this finding even in school children. So in young children, they also have a greater number of dental caries if they have these genetic variants that predispose them to consume sugars. Another study replicated this also in another country, and also in an older population in Italy, also replicated. So important, of course, for us to see this replication because it demonstrates the robustness of this finding. And the take-home message really is, if you have these genetic variants that predispose you to over-consuming sugar, and in some cases with the GLUT2, it's considered really mindless eating because most individuals are not aware that they're actually over-consuming sugar. And it turns out they are consuming an excess amount of sugar almost every day, around 30 grams more of sugar, and that's ultimately having some adverse health effects. Now, just shifting gears a little and looking back at why I think it's important, in fact, essential, that we consider genetics in every study that we do in the field of nutrition. It's because if we trace the path from virtually any nutritional factor to any health outcome, if there have been enough observational studies, we tend to see this heterogeneity of response, inconsistencies among studies, right? How many times do we hear that, you know, a study shows that coffee is good for us, another day a study will say coffee is bad for us, and another day a study will come out and say coffee doesn't do anything? Now, there's a lot of reasons for those inconsistencies, but an important consideration, of course, is the genetics of the population that's being studied. So I've tried to illustrate in kind of a black box 3 possible genotypes. And if we can use that information to predict if a particular nutritional intervention will improve our health, that's wonderful. If it doesn't have any effect, well, perhaps, you know, that's okay as well. But what about cases where the nutrition advice that we're giving might actually cause harm, right? Some people who go on a low sodium diet, their blood pressure actually goes up. Some people who cut back on their fat intake, their blood lipids go up. So it goes in the opposite direction. In other words, in some individuals, the advice that we're giving might actually be harmful. So if there's a way for us to identify those individuals and seek alternate strategies, that's obviously something that we want to do. So clearly, one size does not fit all. And we've known this for quite a long time. In fact, the earliest references to this comes from the 1st century BC, where Lucretius noted that one man's food is another man's poison. What's good for one person might actually be harmful for somebody else. Now, we've come a long way, and certainly with modern tools of molecular genetics and genomics, we're now able to pinpoint specific regions of the genome that can help us understand why we look and function differently from one another. So there's all kinds of different genetic variants. Single nucleotide polymorphisms, or SNPs, are the most abundant and the most well characterized. And a lot of SNPs can explain differences in various phenotypes or traits. Why one person has blue eyes versus brown eyes, or straight hair versus curly hair. Now, you can imagine if these genetic variants impact the way that we look on the outside, consider what they could be doing to us on the inside. In other words, the way they affect how we metabolize everything that we ingest in terms of our diet. Now, I do want to make an important distinction between the different kinds of genetic variants, because this, I think, has caused some confusion in the field of genetic testing. And I want to differentiate between, the so-called disease risk genes or disease-causing genes versus modifier genes. So disease-causing genes are the ones that are really high penetrance genes, things like, for cystic fibrosis. If you have this mutation, you will develop this disease, and those are diagnostic. Now, since the first draft sequence of the human genome was completed, there was a lot of interest and certainly a lot of GWAS studies that sought to identify disease-associated genes. But those have been largely unsuccessful for common forms of chronic disease. They've been very good for identifying rare diseases, so those rare high penetrance genes. But when it comes to common forms of heart disease, cancer, diabetes, osteoporosis, no single genetic variant can explain very much of the variance when it comes to the common forms of disease. So where I think there's great opportunity is really in the modifier genes. So these are genes that alone do not impact the risk of disease directly, but they modify the effects of environment, namely nutrition. So in this domain, we can think of genes that affect the absorption, distribution, metabolism, uptake, and elimination, also targets. So for those of you who perhaps are familiar with the field of pharmacogenetics and pharmacogenomics, you're familiar with the concept of ADME. This principle applies equally to nutrition, as well as toxicology and other disciplines as well. And this is really where there's an opportunity for actionable recommendations. Information that tells an individual, because of these genetic variants that you have, here is what you can do to mitigate your risk. We're not telling people you have a higher risk of heart disease or higher risk of diabetes, because there is no evidence to support any of those studies that simply link common genetic variants to common risk of disease without taking into account, of course, the major effect of environment, namely diet. So when we look to see where and how genes can affect our response, as I mentioned, genes can influence our likes and dislikes. They can also influence our appetite. So some people need more of a particular macronutrient to get the same satiating effect. And once a food is ingested, there are various metabolic pathways and processes that are impacted that ultimately determine how much of the nutrient reaches the target cell of interest. Well, one of the areas that we were quite interested in was cardiometabolic disease, mainly because it is the #1 cause of death, not only in the United States, but certainly in Canada and many other parts of the world. And this article in JAMA a number of years ago identified diet as being responsible for approximately half of all cardiometabolic deaths. What wasn't clear, of course, is exactly what aspect of the diet is responsible, and more importantly, in which individuals. So we were interested in coffee as one specific component of the diet that has been linked to cardiovascular disease. But the studies that have been conducted to date, and there have been dozens, have really been inconsistent. And so we wondered if certain genetic variants might explain some of these inconsistencies. Now, when you look at a cup of coffee, it's a fairly complex beverage, all kinds of bioactives, things like the diterpenoids that are known to raise LDL cholesterol, other things like the polyphenols that have antioxidant properties might actually be beneficial. So it's a mixture of some good things and not so good things. We were particularly interested in caffeine, which is the most widely studied constituent of coffee, and asked the question, is caffeine a specific component in coffee that might trigger a heart attack? This is just the chemical structure of caffeine, 1,3,7-trimethylxanthine. It's the most widely consumed stimulant in the world. Many of us have obviously acquired a liking for caffeine. When you look at how caffeine is metabolized, it's broken down in the liver almost exclusively by CYP1A2, this enzyme that converts caffeine into a more water-soluble compound called paraxanthine. Then that's more rapidly broken down into other metabolites and eliminated from the urine. So CYP1A2 catalyzes the rate-limiting detoxification of caffeine. And the nice thing about the CYP1A2 gene is there's a common polymorphism at a position with this RS number that has a profound effect on enzyme activity or inducibility, as shown here in the y-axis. So if you have the AA genotype, you have a 4-fold higher rate of breaking down caffeine compared to a carrier of the C allele. So we reasoned that if caffeine is a component in coffee that might trigger a heart attack, then we would expect the slow metabolizers to be at a higher risk, right? Because they're less able to get rid of the caffeine. It sticks around in their system longer. It's more likely to have effects on the cardiovascular system. Whereas the fast metabolizers, they get rid of it very quickly. There's virtually no caffeine left in their system. So we conducted this study. This was in collaboration with Hanya Campos at the Harvard School of Public Health, who's based in Costa Rica. And we reported this in JAMA back in 2006. And this was work that was done by one of my first PhD students, Marilyn Cornelis, who's now a professor at Northwestern University in Chicago. So before taking genetics into account, when we looked at the link between coffee consumption and myocardial infarction or a heart attack, we found that those who drank 4 or more cups a day had a 36% increased risk of a heart attack. Now, the real question we wanted to ask was, is this increased risk due to caffeine? And if it is due to caffeine, then we would expect the slow metabolizers to be at a higher risk. So when we stratified our population by CYP1A2, that's exactly what we found. On the left-hand side are the fast metabolizers, and you now see a protective effect of moderate consumption. Whereas the slow metabolizers, even as little as 2 to 3 cups a day, was now linked with a significantly higher risk of a heart attack. Now, I just want to point out that the only difference between these 4 cup a day drinkers on the right and these 4 cup a day drinkers on the left is a single nucleotide polymorphism that affects the rate of caffeine elimination and detoxification. They smoke the same, physical activity levels are the same. All those confounders are equally distributed between those 2 groups. So because caffeine is the only major substance in coffee that's detoxified by CYP1A2, this strongly implicates caffeine as a trigger for a heart attack. So when we reported this back in 2006, a few years later, this group in Italy essentially replicated our findings, but looked at risk of hypertension, which we had postulated was their intermediary effect. And even though the cup sizes are a little bit different and the groups are slightly different, nevertheless, you see the similar pattern. On the left-hand side, fast metabolizers are protected, slow metabolizers, higher risk. This group also showed the same phenomenon with impaired fasting glucose, where they showed that coffee increases the risk of impaired fasting glucose only in the slow metabolizers. Another replication by a group in Brazil showed the same effect. In addition to looking at physical activity as a modifier, they also showed that CYP1A2 modifies the effects of caffeine on blood pressure response. Again, adverse effects only in the slow metabolizers, not the fast metabolizers. All right, now I just want to, again, switch gears one more time to give another example of a different kind of gene-diet interaction, and that comes in the field of obesity and weight management. Now, in this case, we're not trying to use genetics to predict obesity, right? I mean, there have been hundreds of studies that have identified hundreds of genetic variants that have small effects on risk of obesity. And at the end of the day, if you want to know if someone is obese, you just have to measure their body fat or have them stand on a scale. The real question is, for someone who is struggling to lose weight, what is the most effective dietary strategy? And can we use genetics to predict the response to weight loss? Well, one of the most compelling studies comes from Luke Hughes' group at Harvard, where they conducted a 2-year randomized controlled clinical trial, the gold standard of scientific evidence. And they compared the effects of a low protein diet to a high protein diet on body fat and body composition. And what you can see here is the effects of the high protein diet compared to the low protein diet depended on which version of the FTO gene a person had. So on the left-hand side, carriers of the T allele, there was no difference in how much body fat they lost, whether they were on a low or high protein diet. In fact, it looks like those on the low protein diet lost a little bit more weight or body fat, but that wasn't statistically meaningful. But take a look at those on the right-hand side, those who have the AA genotype. those individuals who were on a high protein diet lost a considerably greater amount of body fat, and they maintained it 2 years later. So this shows just how solid this effect is. In fact, this phenomenon has since been replicated. This was a study that was done in Spain, and it showed a similar effect, that a high protein diet only had effects in those who had the AA genotype, the risk variant. This other study published just a couple of years ago in 2019 in the United States also showed the same effect, even in as little as 4 weeks. So they conducted this 4-week hypocaloric weight loss trial, comparing the effects of high protein to low protein, and they found that only those who had the risk variant saw a response with the high protein diet, and again, in as little as 4 weeks. My lab also sought to replicate this using a cross-sectional study. So we weren't able to look at weight loss, but we were able to look at body fat in a cross-sectional study design. So this was work that was done by a former undergraduate student, David Merritt, and a former PhD student, Joseph. And here we looked at a group of East Asians. And in those individuals, waist circumference is a stronger predictor of cardiometabolic disease than BMI. And so when we looked at waist circumference, remember those who have the AA genotype, those are the ones that benefit the most from going on a high protein diet. And sure enough, you can see this here, because in those individuals who were not on a high protein diet, they were on the low protein diet, they had a much greater waist circumference, approximately 10 centimeters. So that's, over, 4 inches greater diameter in terms of their, not diameter, waist circumference. Whereas those who have either the AT or TT genotype, there was no difference in whether or not they were on a low or a high protein diet. So the last example then of a gene-diet interaction that I want to give is some work that we also did a number of years ago. This was work that was done by 2 former PhD students, Leah Cahill, who's now a professor at McGill University in Montreal, in Dalhousie, actually, Nova Scotia, and Benedict, who is a professor at the University of Ottawa. And here, this was part of Leah's doctoral thesis, where we wanted to understand why some people have a different blood response to vitamin C. And so circulating levels of ascorbic acid are an indicator of vitamin C status. And it turns out that this glutathione S transferase is responsible for the recycling of dehydroascorbic acid back into ascorbic acid. So we reported this in the American Journal of Clinical Nutrition, where we looked at different ethnic groups. And first looking at those of European descent or Caucasians, those individuals who have the null genotype, in other words, they do not have a functioning enzyme caused by this polymorphism, their blood levels or serum levels of ascorbic acid plummet if they're not getting adequate amounts of vitamin C. In other words, they're not meeting the recommended daily allowance, or the RDA. But look at those who have the functioning version of this genotype. Even if they're not getting enough vitamin C in their diet, they're still able to preserve what little vitamin C they're consuming. We also replicated this in a group of East Asians and saw the same pattern. Again, on the left-hand side, those who have the null genotype, you see their blood levels drop. Those who have the functioning genotype, their blood levels are preserved. Now, what's interesting and unique in terms of differences between the Caucasians and East Asians is just how common the null genotype is. It's found in only about 20% of Caucasians, but roughly half of East Asians have this. Now, in terms of relevance for a population, this just means that perhaps this polymorphism impacts a greater proportion of individuals who are East Asians. But at the end of the day, it really doesn't matter what your ethnicity is, because it shows that if you're looking at the right polymorphism that has a functional effect, it doesn't matter what the ancestry is. Now, this is something that often, perhaps rubs genetic epidemiologists the wrong way, because they always argue, no, well, the genetic architecture of our population is very different than your population. And so we have to do these studies in all these different ethnic groups. And sure, that's good to do that for replication. But if you're looking at the functional significance of a polymorphism, here's just an example of it really doesn't matter. It's the same thing with the CYP1A2 polymorphism that's also been replicated in distinct populations. That enzyme breaks down caffeine, and that polymorphism has a functional effect on enzyme activity. It doesn't matter if it's in an African, an Asian, a European, or a South American. It's going to have the same functional effect. What is different is just how common those polymorphisms are. But here's an example where it's more common in East Asians and in Caucasians, but still as an individual, I would still need to know, do I have the null genotype or do I have the functional genotype? And it doesn't matter if, only 10% of people of my ethnicity have that, I could be part of that 10%. So you still need to know as an individual. All right. Now, one of the questions that I often would receive is, well, what happens when you give people their genetic information? You tell them what their genotype is for CYP1A2 or GST or FTO. What are they going to do with that information? Does that actually motivate them to change anything? Well, we thought that's an interesting question and one that had never before been tested. So we decided to test this hypothesis. And we reason that, on the one hand, some people might say, yes, I do have that gene. This is going to motivate me to eat a healthier diet. The flip side, of course, could be this, where someone might say, I have the gene. There's nothing I can do. Pass me the potato chips and remote and leave me alone. So this concept of genetic determinism or genetic fatalism, that it's in my genes, I can't change them. There's nothing for me to do. Now, this concept really applies to disease-associated genes. If you tell someone, you have the gene for Alzheimer's disease, and other than just generic advice of eating better and getting more exercise and doing word puzzles, there's really nothing you can do. You're going to have a higher risk of Alzheimer's. That's different than giving someone information on a modifier gene. If you tell someone you have the gene for salt-sensitive hypertension, you need to lower your sodium intake, that's something that they can do. And there's actually action that they can take. So we decided to put this to the test, and we conducted a randomized controlled clinical trial, again, the gold standard. And this was work that was done by a former PhD student. Diva is the student who's a professor at McGill University now in Montreal. And as part of her doctoral thesis, she looked to compare the effects of DNA-based dietary advice compared to standard recommendations. And we found that those who got the DNA-based dietary advice actually understood the information more, they wanted to learn more, and they were more motivated to change their eating habits. Now, motivation is one thing. Did they actually change their eating habits? We reported this in PLOS One that yes, in fact, they did. So those who got the DNA-based dietary advice changed their eating habits. They followed the recommendations to a greater extent, and they maintained it 1 year later. Importantly, it was the right group of individuals, those who had the risk variant for salt-sensitive hypertension. They lowered their sodium intake by almost 300 milligrams per day and maintained that a year later. Now, that was the first study of its kind. Other studies have since replicated this. And because of that, a group conducted a systematic review on studies that looked at genetics and eating behaviors. And they concluded that in those studies where you're not just giving genetics, but you're coupling that with actionable information, those are the studies that result in the most meaningful changes to behavior. So the same group conducted a clinical trial also using our Nutrigenomics test. So the 70 gene test that we offer through our company, this was used to test the effects of a weight loss trial using the diabetes prevention program, or the group lifestyle balance as it's called now, which is the gold standard for weight loss among diabetic patients. And they found that those who got the genetic test on top of the group lifestyle balance, they had the greatest improvements in diet. And they also looked at changes in body composition, and looking at both the 3 and 6 month trial, those who got the DNA-based dietary advice, the personalized advice, saw a greater reduction in body composition, body fat, compared to the standard, the gold standard, which is the general advice by the Diabetes Prevention Program. So this is an example of a fairly hard outcome that demonstrates that giving people their genetic results with specific recommendations for improving their diet on top of the gold standard recommendation actually leads to greater clinical outcomes. Now, as I mentioned to the group of students yesterday on the talk, the field is not without its skeptics. And oftentimes this comes from those who are not familiar with the research approach in the field of nutrigenetics and nutrigenomics. And I find that a lot of these comments tend to come from those in the field of genetics or genetic epidemiologists who ideally would like to have explained everything simply using genetics. And one of the criticisms that I've heard from them is that single SNPs are useless. You have to use genetic risk scores. Well, again, this really comes from population geneticists who tried to identify SNPs that were linked to things like diabetes, obesity, heart disease, and those studies, those large GWAS studies, were just a failure. I mean, I don't want to say a failure. They showed us that common SNPs cannot be used to predict these traits. And so what they ended up doing is looking at several SNPs and creating a genetic risk score. And then they conclude that, okay, we can now, not just 2% of the variation, we can now explain 6% of the variation using, 30 SNPs. Well, that's still not really effective. And because all of these studies really fail to take into account the role of environment, namely diet. So when it comes to the common forms of chronic diseases, yes, you cannot use a single SNP to predict obesity, to predict heart disease, or to predict cancer. But single SNPs, if you look at the right SNPs, they can be very effective in looking at how they modify the effects of an environmental factor. So I've given a number of examples now: CYP1A2 and caffeine, FTO and protein, GSTT and vitamin C, and the list goes on, where if you look at the right SNPs that have a functional consequence, they can provide you with meaningful information on how to modify the risk based on diet. Some argue, well, people aren't even going to change their behaviors. It doesn't matter what you give them or what you tell them. Well, we and others have shown that if you give people the right kind of genetic information coupled with the right advice, they will in fact change their behaviors and they'll maintain it. Some argue that we need more evidence, that this is not ready for prime time. We should not be marketing genetic tests for personalized nutrition because we need evidence and we need it from randomized controlled clinical trials. Well, what's interesting is the vast majority of nutrition recommendations that we give are not actually based on evidence from RCTs like we use in to administer drugs. But it turns out we do have evidence, and we do have them from randomized controlled trials now showing that DNA-based dietary recommendations are superior to the generic one-size-fits-all recommendations. Some argue, oh, it's all about the microbiome. This is the hot area now, and our host genetics, that doesn't matter. It's all about the gut bacteria. We have more microbial cells than we have mammalian cells. This is the future. Well, I don't discount the importance of the microbiome, and I think we're learning quite a lot, but we've only begun to scratch the surface in terms of understanding the impact of the microbiome and its ability to predict response to diet. In fact, the microbiome changes quite significantly based on what you ate just a few days ago. So does that mean you have to measure someone's microbiome again a few days later and say, okay, well, now we got to change you and put you on a different diet? But what's even more interesting is that the microbiome is largely determined by our genetics, the host genetics. In fact, this GWAS study that was done by some of my colleagues here at the University of Toronto identified a number of host genetic variants that can predict the microbial composition in the gut. This was published in Nature Genetics back in 2016. And just last year, another research group also conducted a GWAS study and identified other genetic variants that also explain why some people have a particular gut microbe profile and others don't. So if you do see studies that link the microbiome to any kind of trait, an important question to ask is it actually due to the bacteria, or is that bacteria a surrogate measure of a host genetic polymorphism? I think the jury is still out, and we still have a lot more work to do before we can tease apart the difference between the microbiome and the host genome. The one thing I will say is that it can only be unidirectional. So the host genome can impact the microbiome, but the microbiome cannot impact our genetic sequence. It can certainly affect gene expression in the host, but it cannot cause polymorphisms. It cannot alter germline genetic variants. So any type of confounder would only work in that one direction. But again, I don't want to leave you with the idea that I think the microbiome is not important. I think it is. It's just that we have a lot more to learn before we can begin to consider the microbiome for personalized nutrition. Some argue genetic tests are too complex. People won't understand them. Well, yeah, if you give someone the raw readout from a sequencer, AC, GTC, and tell them, good luck with that, of course they're not going to understand that. But if you tell someone you're a slow metabolizer, you need to limit your caffeine intake, that's not very complex. Some argue that family history is more informative. Well, maybe once upon a time, but families share more than just common genetics. They also share a common environment. And usually family history only tells us something about early onset of disease. But when it comes to common forms of common chronic diseases, family history is really much too crude of a measure. And if you had, grandparents on both sides that maybe had heart disease in their early 60s, it doesn't tell you what you can do about that. Does that mean you need to lower your sodium? Does that mean you should cut back on caffeine? Does that mean you need to increase your folate consumption? There's so many different pathways. And when it comes to nutrition and lifestyle intervention, it's important to understand which of the modifier genes are the ones that you need to focus on the most. And the last, and one of my favorite criticisms is, well, we don't need fancy genetic testing, just follow recommendations for healthy eating. Well, if you do that for coffee and you're part of the 50% of the population that is a slow metabolizer, and you follow the recommendations for healthy eating, which is up to 4 cups of coffee a day, that's what Health Canada and the US Institute of Medicine recommend, that's going to increase your risk of heart attack, hypertension, prediabetes, kidney disease, and all kinds of other ailments. So just following the recommendations is actually not good enough. In fact, it can actually be harmful for some individuals. So after having gone through a number of those critiques that I've heard over the years that, in my opinion, are clearly unfounded, a colleague and I, Bibiana Garcia-Bailo, who again was actually one of my former PhD students, but is currently an adjunct professor at the University of Toronto, and she's currently the director of research and development for the company Nutrigenomics. We published this paper in Current Opinions in Clinical Nutrition and Metabolic Care, where we identified those comments. So in addition to some of the advances, we also talked about some of those controversies that tend to come up, which are really unfounded, and address them head on so that anytime some of these issues are come up, particularly among some genetic epidemiologists, we just send them the paper and say, here, we've already addressed this. So where are we today? Well, we need to eat. I know myself, it's around lunchtime for me, so I'm getting kind of hungry. We currently give dietary advice for healthy eating, but that advice is based on what I call old science. Recommendations are based on studies from 15, 20 years ago. So how much more evidence do we need before we can tell people, all right, based on your genotype for this, here's what the recommendations are. And for someone else, perhaps we need to alter those recommendations. So how do we balance the advice that we give to individuals compared to public health recommendations? Well, there's all kinds of genetic tests on the market. Some of you are probably familiar with things like 23andMe. Some of these other companies have actually gone bankrupt. So MyGene in the bottom right from Australia is no longer operational. Interleukin Genetics and Navigenics in the US, they're also no longer operational. But there continue to be, for every company that closes, 2 new companies will come up. So I just want to briefly mention that we established Nutrigenomics as a University of Toronto startup company just over 10 years ago. So those are really considered the prehistoric days of genetic testing. And it was the first genetic test in the world to be available only to healthcare professionals, so not direct to consumer. So you can't just go to the website and we don't collect personal information and then send you back a kit. It has to be done through a clinic. And there are currently over 10,000 clinics and healthcare professionals, dietitians, physicians, other nutrition professionals. We also have distributors in several countries. Mexico is one of them, of course. The Cleveland Clinic we're their exclusive provider of genetic testing. They've looked at other genetic tests and they've realized that ours is the one that's based on solid scientific evidence, the only test that's been replicated and validated using clinical trials. So our head office is in Toronto, but we have an office in the US, Australia, the UK, and Brazil. We've funded research, not only our own research at the University of Toronto, but research from other universities around the world, and so far invested almost $500,000 in research dollars. Our reports are available in several different languages. Of course, Spanish is one of them. And this is just the cover of a sample health report, personalized nutrition and fitness report. When we first established the company and launched in 2012, it consisted just of a 7-gene test. 2 years later, we came out with a 6-gene test for gluten intolerance, and then a more comprehensive 45-gene test. And our latest test is a 70-gene comprehensive test, and this is the one that we currently have based on the scientific evidence to date. We also have specialized reports for those who are interested in athletic performance, focus on fertility outcomes, weight management, and also a plant-based report. For those who are vegans or vegetarians, obviously their genetics is not different, but their food options are. So we want to respect those personal choices and gave them some options for eating according to their genetics on a plant-based diet. So back in 2007, I was at the first International Nutrigenomics Conference, and a reporter wrote about this and had this headline the following day, how genetics may define diets of the future. And they talked about how this is such a futuristic concept. But if you fast forward 10 years later, in the Wall Street Journal, I was also interviewed for this article, and they talked about not only the research that we're doing at the University of Toronto, but also the company, and how this is no longer something that's futuristic. The testing services are available now, and the science has grown tremendously. And we could now give some people recommendations based on their genetics and not just generic one-size-fits-all advice. So I've also worked with the American Nutrition Association. 2 years ago, we published this position statement that highlighted really the areas that need to be focused on, both in terms of research as well as clinical practice. And so how do we translate? How do we set certain standards? Because the field of genetic testing is largely unregulated, and anyone can just set up a company and make any kind of advice. We have to make sure that the recommendations that are being given are based on robust scientific evidence. So for those who are interested in learning more about nutritional genomics, we also work with the Dietitians of Canada to create this 6-hour course. And they were able to create this promo code. This was really meant for the students, but anyone can actually take this 6-hour course and you get a certificate of completion. You don't have to be a dietitian, and you don't have to be a member of Dietitians of Canada. This is just a way of learning more about some of the broader topics and some of the social, ethical, and legal aspects of genetic testing and privacy and those sorts of issues. And that's available. We also created a much larger 46-hour course through the American Nutrition Association. And again, there's an opportunity to get accredited and get some certifications for that. So if you're interested in that, I will leave my email address at the end. But I just want to finish off by thanking our various sources of funding and our collaborators and former and current students who made all this work possible. And I just want to leave you with this quote. Even though the term nutrigenomics is fairly new, the concept is quite ancient. In fact, Hippocrates noted over 2,500 years ago that positive health requires a knowledge of man's primary constitution. That's really just an old-fashioned way of saying genotype. And then he went on to say the powers of various foods, both those natural to them and those resulting from human skill. And clearly, I think he was referring to personalized nutrition. So I will stop here. Thank you for your attention. I'm happy to take questions, and I will look at the chat box, and also for those of you who'd like to perhaps unmute your camera or mic and ask a question live. If I don't, for whatever reason, get to all of the questions, this is my email address. Feel free to send me an email, and I would be happy to respond. And thank you for your attention.
Okay, thank you very much, Ahmed. So it was very interesting talk. So now the lecture is open for questions. There are some questions already in the chat, and also I can see some hands in the but I don't know, maybe the first ones were from in the chat, I think.
Yeah, so I'm happy to take those questions first quickly, just the first four that I saw pop up here. So a question, I think, from a student yesterday, Miguel, who asks, they're left wondering about what's the process to select the SNPs tested in the randomized clinical trial. So that's a great question. So the one clinical trial that was published in the British Medical Journal, they actually used the Nutrigenomics report. So it was based on that 70 gene test. Now, the question is, what was the rationale for selecting the SNPs that went into our test? And that is a question that often comes up. And really, that's just based on our knowledge of the science. I've been in this field for, going on a quarter of a century now, and I'm familiar with the types of studies that have been conducted and the quality of the studies. We also have an international science advisory board who, between us and them, they're the ones that produce the vast majority of the research studies in this field. So we're familiar with which SNPs can be used and which ones cannot. We've published a large number of null findings showing that certain SNPs are not effective. And I've been amazed to see how some of those SNPs make it into other genetic testing companies' reports. And I know the science because, we generated that science. They cite our paper on their company website, but the recommendations are not at all based on what the actual science shows. So again, I think it really depends on knowledge and expertise of those who are actively doing research in the field. Another question is from Enrique. 23andMe predicts risk of diabetes based on SNPs. Based on your findings, it is likely that some of those variants could be involved in the sugar intake rather than the intrinsic metabolism leading to diabetes. So I just want to say that all those disease-associated risks that you get from 23andMe, you can't rely on any of those. In fact, I know someone personally who got their results from 23andMe, and they were told you do not have the gene for celiac disease. Meanwhile, they were still having a lot of issues, and they did our Nutrigenomics test, which looks at 6 polymorphisms, not just the 2 SNPs for celiac disease. And not only did they have the risk variant, they had the highest risk. So then their physician ordered a blood test, and their antibodies, the TTG antibodies for celiac disease, were 150. They were through the roof. So it shows that the markers that are offered on these consumer genetic testing companies, these direct to consumer, I wouldn't even ask if they're associated with sugar consumption or diabetes or indirectly through environment. They're just not reliable. In fact, for some of the traits, like I think colon cancer or prostate cancer, I have results that says, oh, you have this one SNP, so you have a lower risk of prostate cancer, but you have this other SNP, so you have a higher risk of prostate cancer. What does it all mean? And what can I do with any of that information? What can I actually change? And this is the difference between those disease-associated genes that I mentioned earlier that we should not rely on whatsoever versus the modifier genes that can tell you, based on this polymorphism, this is exactly what you can do. You can cut back on caffeine. You can increase your vitamin D, lower your sodium, increase your ascorbic acid. So another question from LGG. I think this.
Oh, that one is not a question. It's just
okay. Yep.
The next one, Luis Covarrubias, is the next question.
Okay. Yep. Question from Luis. Any effort to track the evolutionary origin of the relevant alleles described? How is the frequency of these alleles in different populations? So thank you for that question. This was actually came up with the students yesterday in terms of, the evolutionary benefits of something like the Glute 2 genetic variant. And as you can imagine, obviously, sugar consumption is important for survival. And once upon a time, those who craved more sugar sought out more sugar from their surroundings. They were able to store that energy for a longer period of time, right? Our DNA doesn't care if we live to be 50 or 60. It just cares that we live old enough to reproduce. And so in many instances, there was a survival benefit, but obviously in times of plenty, some of those variants are now considered deleterious because they're increasing our risk of obesity. But when you do look at the risk variants in different populations, we do see that it is quite varied. In fact, the Glute 2 that predicts higher sugar consumption is almost non-existent in East Asians for whatever reason. So it is quite interesting to be able to track the frequencies of these risk variants and look at kind of the evolutionary story. Question from Patricia. Do you know the effects of the Snip on Glute 2 on brain performance and glucose intake is limited? Oh, if glucose intake is limited. So there haven't been that many functional studies. We do know that mice who have the Glute 2 gene knocked out, they are completely insensitive to glucose. So in Glute 2 knockout mice, if you inject glucose ICV into the brain, they continue to eat. Whereas wild type mice, if you inject glucose ICV in the brain, they stop eating because glucose is sensed by the Glute 2 transporter, and it tells the brain, okay, I've had enough energy, it's time to stop. Whereas when it's deleted, it doesn't have that signal. So we think that those who have the polymorphism probably have not quite a knockout, but something similar in terms of impaired glucose sensing in the brain. But there's a lot more, functional work that can be done that would help us better understand the functional significance of that Glute 2 variant. A question from Esperanza. What is the expected number of genes that should be in a more comprehensive screening? How many more are coming in the near future? So yeah, if you're referring to our the company's report, the Nutrigenomics, we currently have 70. And I've heard others, criticize that and say, well, 23andMe offers hundreds or thousands of genetic variants. Well, we don't have thousands of recommendations to even give someone, let alone hundreds. So, it really depends on the robustness of the evidence. And so we try to keep track in terms of, the advances in the field. But it's difficult to say really how many more. I think what we need to do is to say, look, here we are today. What is the best scientific evidence and how many markers are effective? I showed you the evolution of nutrigenomics, and 10 years ago, the answer would have been 7 genes. This is where we have the most robust evidence. 4 years later, I would have said 45 genes. Today, my answer is 70 genes. There are studies that keep coming out, get published every day. Not all of them are showing us or altering the recommendations that we're currently giving. It is quite possible that as the science continues to grow, we might be able to add additional genetic markers to provide even greater precision, but we're not quite there yet.
So there is one question from YouTube. Can you see it? Ahmed?
No, I just see
Well,
Vanessa's hand up.
okay, maybe Vanessa, go ahead.
Thank you. Thank you for the wonderful talk. I come from the field of pharmacogenomics, and I would like to ask, how does Nutrigenomics compares to pharmacogenomics in the sense that clinical validity and clinical utility are needed for pharmacogenomics implementation? How does that compare to Nutrigenomics right now?
Yes, thank you for that question. A great question, one that we have discussed. Keeping in mind, though, that an important distinction is that everyone needs to eat and everyone needs to eat every day, whereas not everyone needs to take medication and not everyone needs to take a specific medication. So in the field of pharmacogenetics, if you're, say, a fast metabolizer and a particular drug is going to be less effective, then that could, there's certain criteria that need to be met in terms of altering the dose or perhaps looking at a different, something in the same class of medication, but is metabolized by a different phase 1 or phase 2 enzyme. So when it comes to nutrition, we give nutrition recommendations for healthy eating. We don't look at it from the perspective of clinical utility because we're not trying to treat a condition, right? This is saying, you do need vitamin C to survive, and we give recommended daily intakes, and that's based on old science. Is that the right dose for a person? So we have to base it on studies that link those biomarkers to heart outcomes like risk of heart disease, risk of cancer, and that usually comes several years down the road. So again, the field of nutrition science is different in that respect because everyone has to eat and they eat every day, and it's not always based on having to treat an existing disease.
Okay.
Okay, thank you. I just want to close with asking if then this is a matter of risk rather than clinical utility, because at the end, we do want to treat obesity, right?
Right, absolutely. And so if that is the outcome, then we have those clinical trials that compared the gold standard, the diabetes prevention program. Everybody got the diabetes prevention program, right? The group lifestyle balance is what it's called now. And that's been shown to result in weight loss. But the group that also got DNA-based dietary advice for personalized nutrition on top of that, they lost even more weight. And so that's evidence from a clinical trial. So you can quantify that the effect and say, yes, in terms of its clinical utility, we should be giving DNA-based recommendations for weight loss.
Okay, thank you.
Thank you.
Okay, I don't know, Ahmed, if you can see the question in the chat from the YouTube, from Daniela Orozco. You can see it, or I can read it for you if it's necessary.
Oh no, I don't see the one from YouTube. So if you can please.
Okay, the question from Daniela Orozco is: there's evidence of environmental pressures such as scare of food affecting variants related to metabolism or food preference. I can repeat it. There is evidence of environmental pressures, such as scare of food, affecting variants related to metabolism or food preferences.
Okay, I'm not sure I'm quite understanding the question, but I mean, in terms of environmental pressures, I mean, there are a number of factors that influence our food preferences. But again, I'm not quite sure I understand what exactly is being asked. Maybe there's a word there I didn't quite hear clearly.
It's about the scarcity of food. If there's some pressure about the scarcity.
Oh, I see. Okay, so yeah, if food just, I mean, in some areas, if there's famine, for example, and food is scarce, that does cause pressure on certain genetic variants. In fact, this has been one hypothesis for why there's a higher risk of obesity in some indigenous populations, because they've had to endure times of famine. And so those who survived were used to surviving because they were able to live on a scarce amount of food. But now, of course, in today's environment of abundance, those genetic variants are no longer desirable. In fact, they're increasing the risk of early onset obesity. So that has been pretty well documented. It's just been very challenging to pinpoint the specific genetic variants. And I know there's been some efforts to do some GWAS studies, but the problem is, who is your control group? And some in the field have argued there is no so-called thrifty gene. But I don't think that's the case. We just haven't found it yet, because it's very difficult to pinpoint it because you don't have the right kind of control group. So certainly the environment and food availability over a generation or two will have selective pressures on certain genetic variants.
Okay, there are several questions more. So there is in the chat, one from Enrique Corona Barrera. I don't know, you can see it?
Yes, the microbiome. So Enrique is asking, microbiome regulates the gene expression, but cannot modify the gene code. How about in the long term? Well, in the long term, again, the microbiome, there's no biologically plausible mechanism by which the microbiome can cause germline alterations. I mean, we're talking about mutations. What could be changed, of course, are epigenetic modifications. And it's certainly quite possible that the microbiome does cause epigenetic modifications to the host. And those, as we know, can be inherited. So there can be some changes that are, as I mentioned, it can be bidirectional. But in terms of understanding the confounders in current association studies that are linking the current microbiome to a health outcome or the host genome to an outcome, those are confounding each other. But it can only confound each other in one direction if you're looking in a cross-sectional way and you're not doing it in multi-generational study. But certainly over generations, it is possible that the microbiome through epigenetic changes could lead to changes in the host, just not the germline sequence.
The next question is from Tamara Lopez. It's also in the chat.
Yes, so Tamara asks, if there's a negative selective pressure towards allelic variants that cause some limitations, intolerance, or greater risk, could personalized dietary advice attenuate this selective pressure by reducing its effects? It is certainly possible, but again, I think when we're dealing with these environmental pressures, we're really looking at multi-generational effects. So it's not really something that I think we can study in a human population. Perhaps, Mario, with Drosophila, I know that's definitely a kind of work that has been done where you can look at those multi-generational effects in that particular experimental model. And that's something that I think could perhaps shed light in the human situation. But I think just using our limited tools, it might be a bit challenging to test that in humans.
Good. Paola Vasquez.
Yes, so Paula asks, thank you for your talk. How do you think we can solve the problem of opposite effects of some genetic variants at a same phenotype, even when we talk of enzyme activity or other functional effects? Yeah, it's a great point. And this is something that comes up because oftentimes these opposite effects are, sometimes they're due to other neighboring genetic variants. So you have what's called genetic epistasis, right? So some other genetic factor can influence the effect of that other loci of interest. But of course, environment And that is something that has been largely ignored. So even the FTO gene and obesity, if you look at those who are either consuming low protein or high protein, you can come to different conclusions. If you're looking at a population where everyone is consuming, say, a low protein diet, you're not going to see the effect of the FTO gene. Whereas if you look at everyone, a population that's consuming high protein diet, all of a sudden you're going to see that gene have a different effect. So I think the major explanation for those opposite effects are, in addition to possibly genetic epistasis, I would say the environment, and namely diet.
Okay.
I see a question then from A.S. Escobedo. With the common framework in Nutrigenomics, how could one make a genomic profile of people with a tendency to have a strong food addiction to fast foods? Well, in our Nutrigenomics report, we do have, in addition to the GLUT2, the glucose transporter that makes people, have a stronger, they have a predilection to sweets. There's also a genetic variant that affects fat taste perception. And there's also a genetic variant in our report that affects other eating behaviors like snacking. So some people, again, based on a variant in the melanocortin 4 gene, they are more likely to eat frequently. So they're snacking more often as opposed to having meals just at separate times during the day. Now, snacking behaviors don't necessarily have to be bad, right? It depends if you're selecting healthy food choices. But for some people who are prone to snacking on unhealthy foods or fast foods, that is something that having that genetic information could be helpful to it. So again, in terms of the Nutrigenomics framework, we have in our report a section on eating habits and eating behaviors. And there's also a section on weight management and another section on cardiometabolic health, another section on vitamin and mineral metabolism, and a section on food intolerance.
Okay. Well, I think that was the last question.
Okay.
Thank you very much, Ahmed. It was a very nice talk. And as you can see, there were a lot of questions. So the people was very interested in the subject. And well, thank you. And I hope to see you in person someday.
Thank you all very much. It's been a great pleasure. And I do hope to visit your beautiful country sometime soon.
You are welcome to come.
Thank you very much.
Thank you. Okay.
Right.
Thank you very much. Thank you to everyone. So next week, we are not going to have a seminar, but in 15 days, we are going to have a seminar from someone that works with genetics. So you will be communicated. Thank you very much. Gracias a todos, and see you soon.